We have Tracy Shinners-Carnelly. I think a lot of people in the room would know Tracy. Tracy's the Vice President of Research Quality and Sustainability at Peak of the Market in Manitoba. She leads the research program at Peak, which focuses on variety evaluation, improved pest management, and optimization of agronomic practices for fresh market potatoes. Tracy also spends time working with growers in the fields and packing houses, supporting activities in these same areas. She's the current chair of the Potato Sustainability Alliance, and she's also vice chairperson of the Fruit and Vegetable Growers of Canada Crop Protection Advisory Group. Tracy obtained her Bachelor of Science of Agriculture from Dal AC and has a PhD from the University of Alberta. And in 2020, she was appointed as an adjunct professor with the Faculty of Agriculture here in over at Dalhousie in Truro. So Tracy's going to talk about combating insecticide resistance in Colorado potato beetles. I know this is a topic that a lot of people have been asking me about the last couple of years, so I was glad to be able to get Tracy to come and talk to us about it.
Thanks, Ryan, and good morning, everybody. Thanks for the invitation, Ryan, and I have to say this is a great venue. I really enjoyed the presentations, but I'm also enjoying watching the horses being jogged out here because we're having a wonderful winter in Manitoba, but it's not yet outdoor track season yet. So it's a sign of things to come for us back in Manitoba. Like Darren did when he started his presentation, he gave a little bit of a disclaimer. I also want to say up front that I am not an entomologist. So what I'm going to share with you today is really a bit of storytelling and some experiences, observations, and questions that I have asked about this terribly aggravating pest in what I've kind of been dealing with in my career of about 24 years now in southern Manitoba. And I always put that disclaimer out there because when I worked with Manitoba agriculture early in my career, I started to get really interested in Colorado potato beetle. And a colleague of mine at the time, who was an entomologist, reminded me frequently that I was a weak entomologist. And he always said, "You remember that, you're a weak entomologist." And I thought, that's okay, I can be a weak entomologist, but I can still be curious and ask questions, and we can learn along the way. So I'm not gonna try and get really, really technical with you today about entomology and beetles, but again, it's a bit of storytelling. What I'm going to share with you is maybe not the situation that you're dealing with here, but I think as I go through some of the things that we've done over the last number of years, there's some learnings that we found in Manitoba that maybe will be applicable to you as you embark upon a new season and continue that ongoing challenge of trying to manage beetles. So what I want to start with is really sharing with you my thoughts and observations as to why I think this secondary title for Colorado potato beetle as the international superpest applies. Talk a little bit about some insecticide strategies past, present, and potentially future. Then talk a little bit about what does resistance look like. I think when we get lab tests back and we see that a population is reduced susceptibility or it's resistant, we know what that lab result is, but what does it really look like in the field and how can we start to be looking for that? And then also talk a little bit about how do we manage in a field situation when we know we have issues with resistance, and then share with you my thoughts on some strategies that you might want to consider. So here we go. Colorado potato beetle, the international superpest. I came across this term a couple of years ago, and it was coined by an entomologist, a very passionate entomologist, who also was very interested in Colorado potato beetle, someone by the name of Andre Aliokin. I'm not sure if any of you have had the opportunity to ever see him present, but he does really have a strong obsession about trying to manage beetles. I love this term, and I think there's a few factors that really contribute to why it's considered the international superpest. For one, these are big insects. You know, you're in the field, you see these things, they're noticeable, and they eat a lot, right? I mean, I'm sure some of you, or if not all of you, have at some point seen what beetles can actually do. My question is, have you ever seen them eat potatoes, like other than foliage? Have you seen them chew down on actual tubers? So yeah, so that picture here on the left, this is actually a red potato after harvest when all the vines and leaves were gone and there was nothing else to eat, so they started to chow down. So it's quite amazing to see how quickly they can actually chew through tubers. So one of the reasons why I'd consider it to be a super pest, its ability to eat and eat a lot. The other reason I think about this as being a super pest is the amount of stages that are in the field and the continual eating that they seem to do, right? If they're a large insect, they've got a high reproduction rate in the field, and in order to make it through those life stages, they eat, and they eat a lot in order to become a big insect. So we look here and we think about the life cycle. Of course, it starts off early in the spring when those adults emerge from their overwintering locations, usually alongside fields, in tree rows or in other places where they're protected. After a little bit of activity, they'll mate, they lay eggs. Those eggs, I'll get my pointer working here correctly. Those egg masses that you'll find on the underside of leaves will hatch. And when they hatch, of course, it's unleashing this army of little eating machines that are going to start to eat. And that first larval instar stage starts to eat. It's going to go through 4 of those stages before they become those big fat pupae that will then bury themselves into the ground and emerge later as adults. Now, in Manitoba, we used to only see one full cycle. Over the last number of years, and with the warming climate that we seem to be experiencing, we will often get into that second life cycle or second stage of beetles. So in some years, if the conditions are conducive, we will see a lot more late season feeding. And of course, I think the main reason why Colorado potato beetle can be considered the super pest is that it has a long history of developing resistance to any insecticide that's ever been thrown at it. Sometimes it takes a little bit longer, but in time it will develop some sort of resistance. And since the 50s, Colorado potato beetle has developed resistance to more than 50 different compounds from all of the major insecticide classes. So we know this. History has shown that this is the case. And I think as we move forward, and hopefully we'll see new technologies, new chemistries come forward to help manage this, we always need to keep in mind that this will likely happen with everything going forward. And that's really trying to play defensive on our perspective. So now to start a little bit with our experience in Manitoba. So prior to the registration of imidacloprid, beetle management relied predominantly or solely on foliar insecticides, right? So this is the way things used to be in the 90s and before that. And looking back at some work that was done in 2002 with just dip testing, at that time, on populations that were sampled and tested in Manitoba, only Admire, or imidacloprid, resulted in 100% mortality of the population. Beetles were resistant to all those other classes, right? This was probably a common scenario in other parts of Canada as well, I would assume. I think it's very fair to say that the introduction of imidacloprid and Admire really revolutionized insect management in potatoes. Like, this wasn't limited to just Colorado potato beetle. It was to how we looked at insect management in general, because going to that treatment at planting time really reduced the need in most cases for foliar insecticides for all kinds of different pests throughout the season. So it really was a game changer. And because of that, there was widespread adoption of that practice. So fast forward at that time, after just over 10 years of use in Manitoba, we had our first population of beetles confirmed to be resistant to Admire. That happened in 2011. So when we talk about how do we characterize beetles when we say they're resistant or have reduced susceptibility, what does that really mean? So the testing that's been happening over the last number of years to really generate this data is based on a test where beetle populations are collected, they're maintained in the lab, potato plants are grown, and the potato plants are treated with insecticide, and then those beetles are allowed to feed on those treated leaves, and then they monitor the results of what happens. So the scale that I have up here on the screen really shows how those terms are rated. So they look at the mortality of that population after they've been subjected to treated potato leaves. If the mortality has been greater than 70%, so basically over 70% die, that population is considered to be susceptible. If the mortality is only in that 30 to 70% range, it's considered to have reduced susceptibility. And if it's less than 30% mortality, that's what's considered resistant. The interesting thing I find about this, though, is in that yellow zone, right? Let's think of it more of like a gray zone. If you use this product on your farm and you only had 50% mortality or 50% control, how would you consider that response? Would you think that's success, right? It's a pretty wide range, 30 to 69%. So it is a very, very large gray area in terms of trying to assess that, and also factor in the fact that every individual beetle in the field may not be responding the same way. So we end up with the potential of a lot of variability and mixed populations with their level of susceptibility. So when we give a little bit of a snapshot here of what happened in those early days of finding resistance in Manitoba, you look at the chart here with these bars. Of course, in 2008 and 9, there were no populations detected other than those that were considered to be susceptible. There was no testing in 2010, but then in 2011, we only looked at 3 populations, but one of them was considered to be resistant. In 2012, 2 populations and 1 was resistant. So overall, these numbers of the number of populations that were sampled were low, but think of it this way, it's kind of a chicken and an egg. How can you test a population for beetles for resistance if they don't exist? So if control is still being achieved in the majority of places, it's really hard to collect those beetles to figure out how they're performing because they've already been killed. So what ended up starting to happen is in places, in field areas where all of a sudden they were starting to see some beetles starting to show up a little bit earlier or maybe withstand some of that control, the target was to test those populations and see what was happening. So again, numbers are low, but it's starting to be that indicator that something was shifting. So this begs the question then, what does resistance really look like in the field? Again, you can take a sample of a population into a lab, into a controlled environment, test them and get a result. But in a field situation, what do you expect when you've got your eyes out there thinking, okay, how do I look for resistance? Well, this chart really summarizes it very well in saying that field level failures are very uncommon. So what's more likely to have happen is that you're going to see a reduction in the duration of control that you're going to get with a neonic that's, in this case, has been the common one that's been used as an ad-planting treatment. So what the chart shows is that in the first early years of using neonics, on the x-axis here you see the years of use are very low, but the duration of control as rated in those cumulative degree days was quite high. And as the length of time and years of use extend out, what you see that duration of control is actually being reduced. So when we found these resistant populations in that 2011, 2012, it really started to drive the conversation. And people are starting to look a little bit more in the field and make some more observations early in the year. 2016 was a little bit more of a turning point for us when we started to see this more frequently popping up. So the 2 pictures that I show here are good examples of the scenario of what was happening in 2016. So this photo here, the one that I already have the red circle on, is what you would hope to see early on. You've got the plants emerging, your overwintering adults have emerged, they've had a few bites, they take those few bites and boom, they're dead. You want to walk the field, you want to look under the plants, you want to see them on their back, legs up, dead. But what we started seeing more and more of in 2016 was these adults that were out and they're feeding and they look like they're doing okay. So then we start the conversation with some people to say, oh, this doesn't look good. Those adults seem like they're doing a little bit of eating. And then some people will say, oh, no, they're supposed to eat. That's what happens. That's how they get the insecticide and that's what kills them. Well, true, but how long should they be able to eat and still do well, right? That's when the questions start coming up. But when you look at this slide, what do you see here, right? Does that look to you like those are adults that are suffering from eating toxic foliage? I'm not going to go through the biology of exactly what's happening here. I think you can all figure it out. But to me, this is an indicator that those adults fed and they were thriving, right? And that's exactly what we started to see happening a little bit more in 2016. In some locations, these overwintering adults came out, they successfully fed, they made it, they laid eggs, and those eggs were hatching and it was giddy up and go time. So 2016 was really that first year where we started to see this happening at a little bit more frequency in fields in some areas of Manitoba. So we jumped from having a lab test in 2011 that said we have resistant population, then we're seeing these more frequent observations in 2016. So then it's driving that conversation, well, now what do? We've got resistance that we're seeing, we're observing it happening in the field. Well, best management practice would say when you have resistance that's been identified with a pest, you stop using that class of chemistry, right? We've all heard that. It's the theme of what Darren talked about in his presentation as well. If you know you've got resistance to a pathogen, you don't go and keep applying that same fungicide it's resistant to. So we think about that in a practical sense with insecticide management, and particularly with the neonics. So you pull that out, then what? Right? You think about it back in 2016, 2017, we didn't have a lot of great options. Then there's the big but. But we realized that the neonics used at planting still have a lot of benefit overall for insect management because they're still working well for flea beetles and leafhoppers and aphids. So what do, right? And we knew that those observations that we were seeing said that the neonic seed treatments still have a place in that program, even though it's going against our best practice. But what do? How do we manage that going forward? So this really led to an industry-wide project that we conducted. We started in 2015 and went through to 2017. We started it in 2015 because we knew the writing was on the wall. We knew that having that lab test back and knowing that we were getting resistance developing, we knew that we needed to start to do some work to help give growers some good information to say, this is coming on your farm, and when you're at the point where you're starting to see those signs of resistance, what do? How do you juggle insect management, knowing that you've got some resistance and limited options? So how do we use the tools that are still registered for use to eke out every last day of control that we can possibly get out of those neonics? Because we didn't have a lot of foliar options to go to, right? When you're about to go into a battle, we had limited ammunition, and how do you best use it? So this was a very large study that was done at our Peak of the Market research site. We've got a very, shall I say, healthy population of Colorado potato beetles at the site. It's a great location, actually, to do beetle work. So it's a spot to do that and be able to really demonstrate good strategies for management. So this is work that I did together with Darren Gibson and Debbie Jones, and it was funded by many stakeholders in the industry. The real objective was to look at evaluating insecticide management strategies using a combination of all those tools that we had available to us, knowing that we had mixed resistance in the population. Just here as a little bit of a you get a sense at the forefront of that photo, those were untreated checks. So by the third week in July, totally defoliated and left with nothing but vines. But the plots in the back were still managing quite well, even though we had a mixed population there. So I'm not going to get into all the details of this trial, but I want to set the stage to give you an idea of what we looked at, so then you have a better understanding of what some of those key messages are at the end. So what we really looked at was all these combinations of the tools that were registered for use at that time. So if you look through that treatment list, we had the active ingredients, the neonics, so Actera and Titan. We had Verimark was in there, and we had the combinations of an in-furrow treatment or seed treatment and the rate ranges that would have been available on those labels at the time. So the strategy was that most of the treatments would be at planting, because that has been the standard in the industry. We did have one treatment, number 14 here, that was no at planting insecticide, so it would only be managed with foliar options, and then of course our untreated check. The other approach that we took is that we were not going to be using any group 4s as foliars from that resistance perspective, and not to do that. Or if we used a group 28 at planting, that would not be an option for the foliars as well. So again, this idea of if you have that group up front, you don't want to use it later as a foliar. So just to give you an idea of what this looks like as we're going through the season, this chart on the bottom is the number, or the days when the plots were assessed to count for beetles and for defoliation. In this particular chart, we're looking at the number of larvae per 10 plants. So the line here, whoops, wrong button. This line that goes up the top, of course, that is our untreated check. So you see how our beetle numbers really ballooned. These other lines are a different treatment, so it's really messy. But I will point out to you that, of course, this first star here is the plot where it was foliar only. So at some point we had to intervene there and put our foliar insecticide on to knock that back. The next series of stars where we put a foliar on was predominantly within that Verimark group. So our experience in Manitoba has been that Verimark as an in-furrow or seed treatment has really never given us a good duration of control as a systemic. So it's not something that's really been picked up as practice in Manitoba. The other treatment that needed some intervention in here was one of the in-furrow treatments at the low rate. The next batch of plots where we saw an increase in larvae were the rest of the in-furrow treatments. So really the trend we were seeing was the in-furrow applications were breaking first. And when I say break, that means we saw that increase in our number of beetles in those trials relative to the other treatments. This is a chart that looks at percent defoliation for that same timing. And an interesting thing that I'll point out here is this line, of course, is our untreated check. So you can see that by the end of August, we were up over 90% defoliation. But for the remainder of these treatments throughout the season, we were able to keep most of those below 30% defoliation. Okay, so the significant part to this, though, is that when we looked at the yields, and I'm not going to get into the detail of that data at all, the yield of the untreated check was significantly less than all other treatments. The other way of saying that is this line, this yield was significantly lower, but there was no significant difference in the yield of all those treatments. So within all those insecticide combinations that had a range of defoliation by August 15th from about 10% up to just 30%, which 30% defoliation in a field situation looks awful, we weren't seeing a significant difference in yield. So keep that in mind. So just to summarize what we found in that trial, again, the Verimark treatments appeared to break by July in all those years. So it never got picked up as a replacement for the neonics. We just did not see that duration of control. From the 3 years, we did see that the neonic treatments provided good control, even though we know we had reduced sensitivity and resistance within the mix of that population at our site. We did need to move forward with doing foliar insecticides, and that was based on looking at the number of beetles and also rating defoliation. The other trend that we saw was that the highest labeled rate resulted in fewer Colorado potato beetles than the lower rates. Of course, this makes sense, right? It's the dose response. The other thing that was interesting from this 3 years of work was that the seed treatment application generally provided longer duration of control compared to the in-furrow treatments. So this was a significant finding at that time because there were growers that were still using in-furrow rather than the seed treatment option. So again, in the spirit of trying to eke out every day of control that you can get, seed treatment was the way to go. I've got an asterisk by this because, of course, there's been lots of regulatory changes to these labels since the time that this work was done. And as a disclaimer on all of this, when you're looking at using any products, especially when we're looking at these insecticides, be sure to be checking the current label for current use patterns. And of course, at the time, the foliar insecticides we used were Delegate and Exeril, and they were still performing very well. So that brings me into this next part about foliar insecticides. So I'm going to ask a question and poll the audience, and I'm not trying to do any, ageism or biasing here, but how many people remember managing Colorado potato beetles solely with foliar insecticides? Less than half, right? So think about this when you're now faced with, really, in many ways, a new generation of agronomists and crop scouts that have to walk a field and look at how do we scout for beetles and what do we have for thresholds and when is go time, right? It's a whole new learning in terms of how do we do that? So we walk a field and we count beetles and we figure out what the stages are, and then we try and use that. Okay, this is my scouting counts, what do I do? Well, what threshold do we use? And how do we apply that staging in the field to align with some of the labels in the label language? So this, to me, is a whole new challenge of how we need to relearn this. Because when growers ask, well, what is the threshold for Colorado potato beetle? In Manitoba, I can confidently say we don't have a current one. So the work that was done on developing an economic threshold for beetles was done decades ago, and it was done on russet Burbank. And we know the industry has evolved so much when it comes to yield expectations and quality expectations. Work that was done 25, 30 years ago on something like this doesn't necessarily apply. Nor is russet Burbank a great variety to look at for beetles when we've got such an assortment of other varieties and different timings in the field and all those factors. So the answer is we don't have an easy one. When I've searched through extension materials and literature back a couple of years ago, I came up with a few thresholds that I found from different locations. So one is for first generation in areas of significant populations, so we're not sure what significant means. I guess you use your own latitude on defining that. 30% egg hatch is usually what's recommended. So I don't know what beetles look like in the field here in Prince Edward Island, but in Manitoba, when I'm walking the field in late June, early July, that's what I can collect in my hand. So I look at that and I don't know what 30% egg hatch means because I've got every stage of the life cycle represented in the palm of my hand. So when you're trying to do good work and be out there and scout and identify the number of beetles in the staging, you've got all that info, how do you apply what is 30% egg hatch? We've gone past it. So one of the things that we've noticed as we have this mixed population with mixed levels of resistance or tolerance, it also means our staging is all over the board. So it's really hard to look at some of these sort of textbook values and get good guidance on what you're supposed to do. Another one that I find very interesting is a threshold that's 25% defoliation before tuber bulking, or 10% in the first half of bulking, and then back to 25% defoliation Later in bulking. Well, that's kind of confusing. So it's, there's no easy buttons when it comes to how to manage this from a foliar side of things. So my main point in this is maybe you have some good thresholds here, but don't assume that the book value on a threshold is a good one. It's going to be a little bit of relearning that and getting your sense of what really is sort of go time for your area based on hopefully some good local recommendations that have been validated. And of course, the application timing for these foliars are very tricky. These newer insecticides, their safety profile is so much better than the old days when we had those older classes of chemistries, but they also rely on more specific timing because they're effective at certain stages. And that's why the timing for application is really difficult. You want to optimize your chance of success, but it's hard to do when you have lots of stages at once. So just a couple thoughts on some of these resistant management practices when you're dealing with these situations. So again, thinking about when you're using an insecticide, it really is a selection event for resistance. Darren alluded to it a little bit in his presentation about fungicides and how fungicide resistance develops. But there are just naturally resistant individuals in a population. It just naturally happens. So every time you're putting on an insecticide, you're potentially selecting for those individuals that are resistant to it, so they're going to live. They'll thrive and do well. And as you continue to apply that one particular insecticide, you're increasing the percentage of that resistant population and decreasing those that are susceptible. So when you mix or rotate and use a different group, you're knocking back that population that's been developing some resistance. So you need to think about that and then really focus on rotating your insecticide groups. When you're using a systemic insecticide at planting, so whether that's a neonic or if 28 is working for you here, you need to think about not using that same group as a foliar in that same crop. And the other thing to think about is consider all of your options and product use history on your farm. So our example in Manitoba was a couple of years ago, we also started to see some reduced susceptibility to delegate and Exarel. So then we felt we were really hooped because resistance to the neonics and then our foliars that were working were starting to wane. Then we realized there's a registration available for abamectin. Now, abamectin, from our knowledge, had never ever been used on beetles in Manitoba, even though it was an old insecticide. So we used it and had tremendous success. It worked really well. So not saying that's a recommendation here for you, but the thought is just make sure you're aware of what all those options might be. For one reason or another, you maybe haven't used it in the past, but as time marches on, factors change, so you need to be thinking about all these options. So the real keys to thinking about a good strategy going forward is focus on a seed treatment and make sure that you're doing what you can to get all the control out of that that's possible. You need to have the combination of foliar insecticides ready to go and make sure they fit into a good resistance management program and keep resistance management BMPs at top of mind. So this is a very busy chart, but this is where it's trying to put into perspective sort of the status of different beetle populations from across Canada. There's a lot of data that's gone into this one particular chart. It really comes from a lot of work that's been done from 2018 to 2023 as part of the national cluster on potato beetles and really looking at resistance across the country. So this is work that was led by Ian Scott and Chandra Moffat. I've got the full reference later on here in my presentation, but this one summary slide really does a good job of summarizing the susceptibility or tolerance of populations in different areas across the country. So on the bottom here, you see this first group of the neonics, and I'll just point to our Manitoba bar here. You'll see that over that time period of those 5 years, about 18 of the populations that were tested, or just under 40% of our populations, were resistant to the neonics. And of course, the majority were in this yellow zone of reduced susceptibility. For you in Prince Edward Island, which was this last bar, you had no red, so that's fantastic. You had no confirmation of neonic resistance in that time, but 10 of the populations that were tested were showing that reduced susceptibility. But again, remember that yellow bar, it's from 30 to 70%. So this is the warning, right? Yellow, like caution, that's really what should be applied. When we look at the spinosins here, in Manitoba, again, we had quite a mix, and you can see that between our combined reduced susceptibility and resistance, this is why we really have to be cautious in what we're doing and how we're managing our foliars. In Prince Edward Island, this was a bar where you've got lots of reduced susceptibility, and you've got one population that was confirmed to be resistant during that time. And then with the diamides, in Manitoba, again, we have a little bit of everything. We've got all colors represented there. And for you in Prince Edward Island, there were 17 of the populations that did have some reduced susceptibility. So what does it all mean, right? So here's a summary of your PEI data during this time. It means that 20% of the PEI populations that were sampled during that time had reduced susceptibility to the neonics. 55% had reduced sensitivity to the spinosins. 1% was resistant to that group, and 35% had reduced susceptibility to the diamides. So this slide really sums up why it's important to be paying attention to what's happening, and hopefully stay in this zone and not slip into the zone where we are in Manitoba, where we have a lot more resistance that we're trying to juggle. So just to start to sum up some of these management strategies and things for you to consider based on that previous slide with your beetle situation here in Manitoba, or in Prince Edward Island. So keep in mind that the neonic seed treatments, they do remain a valuable tool for insect management in potatoes, but keep in mind that the efficacy is declining on Colorado potato beetle, right? So keep your eyes open for that. And you need to look for those signs of reduced susceptibility. Don't just rely on the data that comes back from a limited number of populations, right? You know best what's been happening on your farm. You know best the insecticide strategy that you've used over the last number of years. And you know best what it looks like when you're walking the field and you're looking under those plants early in the year, or you're peeking around and you're pausing for a little bit longer to watch those beetles and see what they're actually doing. Are they continuing to chew and thrive, or are they showing signs that they're twitching and on their way out? These are all going to be important for you to stay ahead of what's happening. Are thresholds available? Think about that. Ask some of your local specialists and find out how well some of those thresholds maybe work. And use foliar insecticides as necessary. But my big caution here is be cautious in how we use that ammunition, right? We still don't have lots of options for foliar insecticides, especially with the rotation that you need to do. And think back to quite a few slides ago now when I showed you about the defoliation and how that related to yield. The reality is that the plants can actually tolerate a lot more defoliation than we can tolerate looking at. Often my first recommendation to a grower when they'll say, oh, I've got some beetles chewing in my field, and I'll say, look away, right? Don't be too quick, because you've only got limited shots and you don't want to be too quick to use it up, okay? So think about that. And then when choosing a foliar, consider those insecticide groups. Again, if you're using something up front at planting, don't use it as a foliar. And be aware of premixes. When you're doing your group watching, there's been some newer products that have come out as a premix, so you've got 2 groups in 1 shot, and you need to be aware of that when you've got limited applications. And of course, rotate, rotate, rotate. So scout after your application, keep good records, and use them to inform your future decisions. just here's a shot of one paper that's been published by Ian Scott and his large group of collaborators that were working on this work. So the chart that I showed you is included in this particular full paper that's been published, so that's available if you want to have a look at it. And also all the cluster reports for the project that happened from 2018 to 2023 can be found at the FEGC website. You can click through there and get a lot more information on those projects. So I'm wrapping up. I see Ryan is giving me the hook here pretty soon, and I have a bad reputation for chat. But I will just wind up by certainly sending out some acknowledgements. The funding agencies and the organizations and people that I have listed here on the screen were really the collaborators for the project that I referred to that we did back in 2015 to 2017. It was quite a large collaborative effort then, so I certainly don't want to forget about the support and interest that we had back at that time in doing this work. And of course, this is also the cluster project and the full title of this project, the lead investigators, and of course the acknowledgement that this was part of the cluster project, which was funded through the Canadian Agricultural Partnership, as well as the industry partners that funded through the Fruit and Vegetable Growers of Canada. So with that, I hope you enjoyed my beetle stories. And if there's time for questions, if not, I'm around for the rest of the morning.
Where's the mic? I just want to make the comment that like a lot of the insecticides that we have now that still work against Colorados are really only effective against the early stages. And so that's a consideration into like how quickly you have to go. And then also, if there's high temperatures, if it's really warm, the egg masses can hatch much more quickly than under cool conditions, and the development of the instars happens more quickly too. So it's, you know, those are two considerations to make when you're looking at the choices we have. There's been a couple have come out recently that will also get the bigger instar and the adults too, but so choices that way are limited.
Yeah, and I think those points you raise are also reasons why it fits into this international superpest, right? When we think about, you know, I've been asked too, how quickly is it going to be that they'll move from this stage to the next stage? Well, the answer is it depends. It depends on how warm it is and how quickly they're going to move through those stages. And again, your point about our newer insecticides and that early stage, exactly why it's a challenge of now managing foliars, because we don't have these nice distinct stages in the field anymore, at least in Manitoba. So when you're going and scouting, you're seeing everything. And it's really hard to figure out that timing. The other point that I will raise is one of the things that we've noticed, and I even forget which year it was, Darren, you may remember, but we've been through a few really dry years in Manitoba. And we had a spring where we had decent moisture, our population woke up, it got going, and we were seeing multiple egg masses on plants, and we thought, boy, are we going to be in for it. And then nothing happened. Nothing happened. Nothing happened. And then we're out there looking and scouting, the eggs all dehydrated, so they never ever hatched. So we think this was, if there's ever such thing as a benefit of a drought, this was another observation we made, is that, you know, we think about them and how they respond to heat, and they go faster in heat, but there was moisture that was also needed to be able to maintain those eggs. And we had such a dry year, they were just popping, just nothing.
Tracy, I'm one of the old guys in the room. I work for Bayer, so I certainly remember what things were like back in the late 80s, early 90s, et cetera. In general, your data seems to show that Manitoba now has a worse resistance problem than the Maritimes for sure, and maybe other areas. And yet, we've probably used Group 4s as heavily as anybody since Admire was launched. Do you have any theories, or from other colleagues you've talked about, why you seem to have a worse problem in Manitoba now than we do in the Far East, where, like I say, we've probably used as much or more than anybody over the years?
So the simple answer is I don't I have a few theories, and I also want to make a little bit of a disclaimer too here in that this isn't an entire Manitoba scenario. I would say that our challenges and most of the data that I showed for Manitoba was based on our experience in south central Manitoba. What we're seeing in other parts of Manitoba now, it's starting to develop a little bit more, but I would say predominantly beetles are in that south area. In that south area, we're also predominantly fresh market area. The size of fields are a little bit different. The likelihood of overwintering sites are a little bit more. So a few theories that we've thought about in terms of why this seems to be happening. I would say one, we're looking a lot is one part of it. The other is, and this is only a thought on my part, in Manitoba, the majority of the crop is harvested green. So the beetles are in the field, and I think some of them are actually getting crunched up and disintegrated as the harvester is coming in there and done. In the fresh acres and areas where our seed is, everything is top killed. So we're top killing, and then we're forcing those beetles out of the field to go look elsewhere, and in a lot of cases, we're promoting them to move into overwintering sites. So they're getting potentially early, and they've got a little bit more carryover. I don't know that for certain, but it's a little bit of a theory. The other thing that's been speculated on, and I don't know this because I wasn't in the industry at the time, but there are thoughts that early on, when imidacloprid was first introduced, that lower rates were being used at that time versus, the full labeled rates because the active was so effective, right? There was good response. So the thought being, well, maybe it was set up that way a little bit in the beginning because we brought on that reduced sensitivity earlier with lower rates. So great question. Don't know the answer, but certainly lots of speculation of why that might be.
Yeah, I'm just wondering, what is the typical crop rotation they use in Manitoba? And one of the things that's different about PEI, we actually have a crop rotation regulated here. You're only supposed to grow potatoes once every 3 years. Mm-hmm. Is there a lot of canola or other crops grown in between? In other words, were there other crops grown where they may have used a neonicotinoid?
So our rotation would be potatoes every 3 or 4 years. So that part is covered. There are other crops that use neonics, but I don't really think there's been, that certainly hasn't been something that's really been part of the speculation as to why that's really happening.